Viewers, Assalamu alaikum and good evening. Today we will be discussing prerequisites in drug development and its translation into clinical practices. You see, most of the research is carried out in basic sciences and very rarely it is translated into clinical practices. Developed countries have developed standards, and the problem of developing country is that they usually carry research in the basic sciences and very rarely they are translated into clinical practices. So the motive for this lecture is to let we educate the target audience, and this is the target audience, you see, particularly in PhD scholars and the regulatory authorities as well. And because sometime we don't have relevant legislation, and that can pose hindrance to those who want to perform independent research. And if there are independent research, then there are certain ethical issues which is not the domain of this lecture, but nevertheless these all are the target audience. Now objective is to find a focus, further focus on alignment of the basic techniques or pharmacological sciences, and let the viewers and reader of these slides know the fundamentals of toxicity studies that usually we ignore in routine pharmacological studies, because we are merely focused on effective dose of the target molecules. So mostly work is done on these on determining the pharmacological screenings and targeted pharmacological activities like antihypertensive, calcium channel blocking activity, or whatsoever the target of anti bacterial activity, or whatsoever the case is. But our findings on toxicological screenings is very rare, and that is very weak. This is the message. So let we educate the viewers that what are the fundamentals of basic toxicity screenings, and at least these sort of screenings are prerequisites before we translate our research into clinical practices. Now, just food for thought, research is carried out globally, but research in the third world countries, research in developing countries, and research in developed countries. Of course, countries like United States and other developed countries like China and United Kingdoms and Japan. I regret to those if I have not named who are developed, but the point is they have worked and they have made relevant legislations, and perhaps they are executing, they are excelling in their respective area. But what is the problem of third world countries? We don't have food, and the developing countries, you see, most of the research is carried out in basic sciences. Rather, we, the developing country, researchers usually research for the developed country. How? Because we perform basic research, we are unable to translate them into clinical practices because we have not enough resources. So we, in fact, research for the developed country. It's just like that we are producing good doctors for the other countries. So having said that, we should have a balanced approach in basic sciences when its implications into the clinical sciences, so that end of the day it should have a trickle down effect for our society. Now, you are quite familiar with the phases of drug development. That is a preclinical phase, which is usually carried out experiments in animals, with a clinical phase, which is usually carried out in human. The preclinical phase comprises on toxicity studies, pharmacological actions, pharmacokinetic, and of course its dosimetry in experimental animal. Whereas In clinical phase, clinical means something around the bed. So you involve the experimentations in the intervention are carried out in human beings, and this is having confirmative pharmacology and then the human biotransformation studies. And once we have data of these sort of data, so then we can easily apply this into a full-fledged clinical trial. Full-fledged clinical trial means it is now registered with relevant regulatory authority, and then we proceed. Uh, what is the message? The message is that we usually focus on clinical trials while our clinicians are not focusing on this sort of study. So 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 what is the message? That if something is going on in the clinical research, it is not preceded by this preclinical phase or the toxicological screening, and we just merely perform the LD50 and nothing more than that. But the story is not up to the determination of LD50 or ED50. This graph is a typical graph that describes the phases of new drug development. The synthesis of an examination of certain molecule, and 1,000 molecules are then screened. So it enters the preclinical phase, and then we just determine that the lead molecule, that is 10 to 20, and then it goes to phase 1, phase 2, and phase 3 clinical trial. While I have not shown a certain legislative acts or conditions that are prerequisites for being processing the lead molecule from preclinical phase into clinical phase. There is a lot of documentation and legislation and regulatory authority involvement. That is not the domain of this presentation. Now you reach to phase 4 clinical trial, and because of the successful completion of phase 3, and this is called post-marketing surveillance phase as well. So usually 10 to 20 molecule goes to up to mature. That gives one molecule is being marketed, that is introduced into the market, and registration is given. But this is a period. So you started from zero period. Basic research is here 1 year till 12 years. So sometime research can progress. That is why it is very costly process, and developed countries have their budget. In Pakistan, we have 1% of the or 2% of the total pharmaceutical business, and that money should be used now on research as well. Uh, Types of toxicity screening, you see. So it is usually toxicology involved the acute toxicity studies, in the chronic studies, and subacute studies. Let me go and see. Therapeutic index is the lethal dose 50 divided by the effective dose 50. We know that. So regarding acute tox study, we determine the LD50 of the molecules. But usually I have been through so many research papers that as an editor, associate editor on different journal, that they just follow this LD50 study in rats or in mice, meaning that it is carried out in rodents. Whereas the prerequisite says that it should be performed in rodents as well as in non-rodents, usually the rabbits as well, because this will encourage you. This is a bit higher mammal, and this is a lower mammal, lower caliber. The BMR of this rodents are high as compared to the BMR of rabbits, and usually sometimes it correlates with human being as well. So in acute toxicity studies, usually we give exponential dosing like 1 milligram, 10 milligram per kg body weight, 100,000 and 2,000 milligram per kg body weight. Sometimes we follow the OECD guidelines, but it's not necessary to just only follow the OECD guidelines. If a country is having its own regulatory authority and they have determined their standards, so we should follow that. Respective scientists should follow that procedure. But nevertheless, what why system is. The why system is that while administering these sort of test dosing, one should be quite vigilant about its effects on different system, like respiratory system, central nervous system, and urinary system. So there are certain parameters available like increased motor activity, tremors, clonic convulsion, tonic extension, stroke reaction, pyroreaction, and there is lengthy list. So we follow it till maximum dose 2000 milligram per kg. And here is an example. Yes, if this pyroreaction is there after 5 minutes, meaning, so you will score like this one. If it is not, it is nil, so it means that the molecule is not producing any these manifestations, signs and symptoms manifestations. This is very important, and these signs and symptoms will be then carried out when we then matched and mapped in a subacute and a chronic toxicity studies. So keep in mind these signs and symptoms. Now the focus is on that we usually focus on effective dose. For example, we are going to screen a molecule and its possibility hypertensive activity. So we make a standard and we compare its possibility hypertensive activity with the standards, and thus then we report it. But we sometimes determine the lethal dose, but we cannot correlate this with the possible signs and symptoms as I referred to in the previous slides. Thus we are doing this work. We need to document these signs and symptoms as well, because if it is a lead molecule, this sort of data will give us very encouragement once you will be submitting our file to the regulatory authority. Now we discussed about acute study. Now we are talking about subacute studies, and again it is carried out in rats and dogs, but the starting dose is the expected animal therapeutic dose, and then increased by 2 to 3 days with a toxic signs symptoms are observed. And remember, these are those toxic signs and symptoms that we observed in acute toxicity study with different dosing. So if the expected animal therapeutic dose is determined, and then you increase the dose by third or fourth day rather, so what happens until the toxic sign are observed? And now this will be a cross check on the previous data as well, and then perform pharmacokinetics for determine area under the curve, half-life, bioavailability, and then maintain the animals per maximum tolerated dose for 2 to 3 weeks as well. These 2 to 3 figures are very important. 2 to 3 days, 2 to 3 weeks, and then you follow up for 2 to 3 weeks, and then the histological studies are performed. You excise different tissues of the vital organs and the connective tissue of course as well, and then determine this tissue penetration and pharmacokinetics, and you get a data. Now regarding chronic toxicity studies, you see there are certain steps. It is carried out in rats and dogs again, but this select a maximum effective dose, check it for the growth retardation and possible pathological changes. This point is important. Select maximum effective dose and select the 3 dose level at least. So one will be the low dose, the other will be the high dose, and the third one will be a midway dose that is fixed somewhere else between. But how it is being selected, this is now explained in the next slide. How to select these 3 dose level. So first select the low dose, but how? It is twice of the expected therapeutic dose. Then select the highest dose in the second dose, and that is 10 times of the expected therapeutic dose. This is important. This is 10 times. And then the midway dose, the mid dose is the third dose, and that is selected midway between the highest and the lowest dose. The highest and the lowest dose, so the midway. But determine the growth retardation and pathological changes. At the end of the day, you will get the data, but you have to note the changes at an interval of 2 weeks. So this body weight, food intake, renal function, hepatic function, hematology, what specs on the CBC, and then the pulse rate, blood pressure, blood levels of compounds as well. So pharmacokinetic studies, plasma level of drugs, and this blood pressure, you see, you must have an advanced telemetry system as well. So it is your lab should be well equipped as well. And then up to 6 months, you see, sacrifice and perform autopsy for all vital organs, especially and determine their weights of the vital organs, examine for graphs, morphological changes and histological changes under microscope. What is important that the long-term studies in men shall be preceded by at least 3 to 12 months of chronic studies in experimental animals. This is important, especially in chronic studies like these drugs that are to be used for months and years, sometimes like hypertension management, for hypertension management, and disease modifying agents like drugs used in the treatment of rheumatoid arthritis. So introducing such sort of drugs, you see, that must be preceded by the chronic study in experimental animals before it is you get an approval for the full-fledged clinical trial to your concerned regulatory authority. So in nutshell summary, the toxicological screening are the basic setup in the new drug development process. Meticulously plan your research activity and make it align with your research projects. Sometimes we focus on publication, but what I suggest, never focus on publication. Focus on the prototype of other, and that how we are going to trickle down for our society. I hope now you understand. Thank you.